Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. This is actually going to be our last meeting of the year of 2023. So thanks, everybody, for your support through this year and through previous years. We've had a lot of really good sessions. And today, we're going to have another great session. And this is actually with a special interest group of ISEV. I think this was actually the first special interest group that was formed, coming together from a previous task force. And this is the special interest group on genitourinary extracellular vesicles. Before I hand it over to our moderator and presenter today, I just want to remind everybody that you will be on mute and with your video off during the presentation, but at the end, we will allow you to share, to ask the questions that you have, and just please put those questions and those comments in the chat box for us, and we'll get to you. So I would like to welcome now Monica to share your screen, and you can please introduce this special interest group, what it's all about, and then move on to the presenters. So over to you, Monica.
Thanks. So I'm just going to give a brief introduction of GUSEV, which is a special interest group on genitourinary system EVs, which includes kidney, bladder, and reproductive organs. We're not biosample specific, so plasma, urinary, and reproductive fluid from genitourinary system are all included. Our goal is to enhance education, cross-disciplinary collaboration, cross-society partnership, and professional networking. Our upcoming activities include the GUSEV Education Day on the 16th of February 2024, program details to come soon, and a symposium at ISEV 2024 in Melbourne. Associate Professor Ute Erdbrugger is the chair from USA, and co-chairs Associate Professor Elena Martens-Uzenova from Netherlands and Associate Professor Dylan Berger from Canada, and I'm from Australia. We invite anyone interested to join via the enrollment form on our website, and this website will be populated with educational resources as well. So today we will discuss some very elegant work on messenger RNA in urinary EVs and how mRNA can be harnessed to stratify risk of kidney function decline and diabetic kidney disease. This area is particularly interesting because most of the RNA work in EVs have been looking at small RNAs, and that's mostly due to difficulties looking at small amounts of long RNAs such as mRNA. So without further ado, I'm going to invite Associate Professor Maija Puhka and Dr. Karina Barreiro and Associate Professor Om Prakash from Institute of Molecular Medicine, Finland, at the University of Helsinki to present their recently published work. Thank you for sharing your work with us.
Okay, so first of all, I would like to thank the organizers, Ken, Monica, ISEV and GUSEV, for the opportunity to present our work. So indeed, we published this paper last April in iScience, and I was thinking to begin with a little more introductions. It's still a bit more introducing the people and the infrastructures behind the paper. So most of the work was done at the Institute for Molecular Medicine of Finland, located in the medical campus of University of Helsinki, in Meilahti, Helsinki, and Finland. And actually, our institute hosts a pretty large biobanking infrastructure, also a very active technology infrastructures, including our HyPrep core, which is specialized in sample preparations, also the thin genomics, where all the sequencing was done. And then it also hosts many molecular medicine research groups. And in our case, this group was funded by the BTKD project that is from EU and under IMI2. Here are the important people behind the paper. So the study was initiated by Leif and Per-Henrik Group and Tiina-Maija Tuomi, who are studying diabetes in the long-term projects called DIREVA and FINDIAN, and they are like have continued for tens of years in Finland. And from there, we got very nice urine sample collections and clinical understanding of the diabetic kidney disease. And then we had Harri Holthöfer, Antti Rannikko, and me myself, who had been studying urinary EVs in many biobank samples. And of course, here today with me, we have Karina Barreiro and Dwivedi, who are the two first authors in the paper and developed these UEV pipelines further. And of course, many others from Helsinki and Lund team. As collaborator, we had also the IB project led by Maria Gomez and also Dennis Delig from Böhringen. And this work wouldn't have been possible without our high-prep core. And here we are developing services and technologies for high-end and high-throughput sample preparations, targeting mainly genomics. And this includes also the EV samples. And here we collaborate with Pia Siljander, who has the EV core facility here at University of Helsinki. So back in 2016, when we started the big DKD project, we were mainly interested on the urinary EVs as liquid biopsies to whether they could replace the traditional tissue biopsies. So it was known that of course we get the most accurate diagnosis of diabetic kidney disease with the tissue biopsy. However, they come with complications such as bleeding, and that carries risks to the patients. So we were interested on this relatively easily available sample source of urinary EVs that would also allow repeated sampling and monitoring of the patients. And we knew that they carry a relatively rich cargo and potential biomarkers. They carry information of the pathological conditions of the kidney, and like it was common knowledge that they carry messages between cells, potentially also within intranephron, and they perform important tasks in waste management. So the most important questions we had was where do urinary EVs come from, which tissues, which cells, and whether the UEV could serve as a liquid kidney biopsy, and whether we could find relevant biomarkers for either early or late diabetic kidney disease. There was, of course, many knowledge gaps. We didn't know many things about the UEV workflows. These were later on actually acknowledged also by the Urine Task Force and published in the position paper from 2021. And we had many questions, starting with which type of urine collection would be good for urine EV transcriptomics, and whether we should add protease inhibitors, which freezer temperatures are fine, and so on. So over the years, we studied many of these things and published our findings also with our collaborators. And actually quite many of these studies were published in this iScience paper. And now actually I would like to give the floor to Karina Barreiro, so she will explain detail of those technical studies.
Hello everyone. So today I will walk you through the technical comparisons that we did in the iScience manuscript. So I will start with pre-analytics. So for this kind of studies, like biomarker discovery studies, we need to work with many samples to have a good statistical power. And in order to do so, sometimes we have to collect the samples from different biobanks or collections. And in order to do that, we need also to know if we can combine those samples in one analysis together. So today I'm going to talk about three pre-analytical factors to take into consideration. One is the type of collection of urine. So this can be, the most common are 24 hours and overnight urine collections, pre-cleaning of urine before freezing, and also to take into consideration the replicability of the workflow. The rest of the points that are mentioned here have been already acknowledged in 2 other publications in the Journal of Extracellular Vesicles, so I'm not going to talk about those today. So first I want to mention some points about our workflow. So from urine samples, we isolate urinary extracellular vesicles by ultracentrifugation, and then we did characterization of those extracellular vesicles that we were isolating. And from the extracellular vesicles, we isolated RNA. And from that RNA, we ran a fragment analyzer, specifically by analyzer picochip, to know the quantity of RNA that we were getting from each isolate, and also to have an idea of the quality of the RNA that we were getting. And afterwards, the samples were sent to messenger RNA sequencing. For the protocol of messenger RNA sequencing, we started with 1 nanogram of RNA, and for the library preparation, we used a SmartSeq ultra low input RNA kit and Nextera XT DNA. And then once that the sequencing was done and we got the reads, we did the read alignment using a STAR algorithm, and then on those reads, we did different quality controls. So with the STAR alignment output, we can know how many of those reads were mapped to the genome and how many didn't. And then with the RCQC, we could study the distribution of those reads over the genome. So how many of those reads were exonic, that are the ones that we are interested in, and then how many map to the introns and the intergenic reads that have to be more with the content of the DNA or contamination of DNA in the samples. And then we studied the sample-to-sample correlation, hierarchical clustering, and for instance, principal component analysis. And before starting with the sequencing results, I want to show you our results on the urinary extracellular characterization. So we did an electron microscopy of our isolates, and we found vesicles of typical morphology, different sizes. The shape was round, and this is a negative staining, so of course we found the typical cup-shaped vesicles found in this technique. And then we did Western blotting, where we found the markers, EV protein markers in our samples. We did also nanoparticle tracking analysis, and combining both the data that we can get from the electron microscopy and nanoparticle tracking analysis, we found out that the population that we were isolating of extracellular vesicles was between 30 and 150 nanometers. And I want to highlight that the protocol that we are using to isolate the extracellular vesicles has been characterized previously in other publications by us. And this is, for instance, one example of our publication in 2021 in the Journal of Extracellular Vesicles, where we also detected different contaminants that, of course, is required. And so we studied calnexin and also the Goldstein marker 130. And well, here we can see again some extracellular vesicle markers. And we also detected THP. THP is a very abundant protein in urine, and it can be, of course, attached to the corona of the extracellular vesicles, but also as a contaminant in the samples in the isolates. And now let's talk about the sequencing results. So first, I want to say a few words about the sequencing quality. So in these plots, we can see the percentage of reads, for instance, in the upper part, percentage of reads map uniquely mapped, and the ones that weren't mapped to the genome. So as we can see, most of the samples had more than 70% of the reads uniquely mapped, and we regard this as a very good quality. And as you can see, the number of unmapped reads, because of the reads that can be too short, and this has to do with the RNA degradation, it's minimal in most of the samples. Then if we see the lower part of the figure, we can observe the reads that were mapped to the exonic part of the genome, and as we can see, it's mainly most of the reads were mapped to the exonic reads. which is very good. It's almost 90%. Of course, this is what we expect from messenger RNA sequencing. But in some cases, when the quality of the samples is not good, this can be different. And something that we can observe also is that the contribution, the percentage of intronic reads and also intergenic reads is minimal. So now, before continuing with the sequencing results, I want to clarify some things that I'm going to mention in the next slides. One is that when I mention robustly commonly expressed urinary extracellular vesicle genes, I'm referring to protein coding genes with at least one count in 90% of the samples. And in our case, this corresponds, or we got with this filter more than 10,000 genes in our samples. And then I'm going to mention also kidney and rich genes. And this is a list that we got from the Human Protein Atlas. There are 413 genes in total. And these are genes that are detected in the kidney. Also, they are detected in other tissues, but they have the characteristic of being elevated in the kidney. And of course, we didn't find the 413 genes in all our samples, but we found more than the half of these genes. So now I wanted to discuss a little bit about the technical replicates and the plateleting step and how these affect the profiles of the RNAs found in extracellular vesicles. So here you can see two expression heat maps and hierarchical clustering. And in the first one, you can see the representation of all the commonly expressed genes. In the second, on your right, the kidney and rich genes. So in both cases, we found that the pairs of technical samples were clustering together. And they show, in other analysis that we did, they show high correlation. So this is telling us that the replicability is good of our workflow, and also that the pre-clearing step is not so crucial, at least in this condition for these samples. Next, I move to the urine collection type. So again, we have the representation of the most commonly expressed genes and the kidney and rich genes. In this case, I want to mention that not all the pairs of 24 hours urine and overnight samples cluster together, but we didn't see neither a systematic difference for this group. What I mean is that it's not that all the samples of 24 hours are clustering together and then the overnight on clustering together in another cluster. So they are not as similar as the others, but they are not so different either. And another thing that I wanted to discuss today is about the sex-driven differences in urinary extracellular vesicles RNA profile. So here we wanted to explore this because it has been described that the physiology and the metabolism of the kidney differs between male and women. So yes, here we had only in this case 3 samples from females, but what we notice is that when we analyze the samples, checking only the sex chromosome genes, that in our samples were 329 genes, the samples from male and from women and men cluster differently. So they are separated. The profile, of course, and this is expected, is different. When we analyze the kidney and rich genes, the samples, women and men samples, are not clustering together. So in this regard, if we check only these genes, the samples, the profiles are more similar. And then when we check the robust expressed genes in the middle, well, here it's an intermediate case, but still the women samples tend to cluster together. Of course, we need to repeat the analysis with more samples, and Maija will tell a little bit more later about our future plans. And last, I want to talk about the RNA quality. Most of the people that have worked with RNA derived, for instance, from tissues or cells and have run the fragment analyzer, they know more or less the profile, how it looks when a sample is degraded and when it's not degraded. In the case of Agilent, they have developed this ring value. So in the case the ring value is 10, the sample has is almost perfect. It doesn't have degraded RNA, and this ring value is based in the ribosomal peaks. When we move towards the ring number 2, the samples are, this indicates that the samples are more degraded. The problem that we have is that when we check the extracellular vesicles RNA profiles, they look closer to what it could be a ring number 2. So it could be completely degraded, but this is because the RNAs in the vesicles is more fragmented. But of course, now we don't have any way to check which RNA is good or bad, the RNA that is coming from the extracellular vesicles. So let's talk about what we found. So typically, when we analyze the RNA in the bioanalyzer picochip, we found these profiles, what I'm showing now. So we have a main peak that is in size less than 200 nucleotides, and sometimes we found some small ribosomal peaks, but sometimes these ribosomal peaks are not there. And this is common from the RNA derived from women's samples and also from men's samples. But when we started to process more of the RNA samples derived from women, we encountered that 27%, more or less 27% of the samples derived from women show a different kind of profile. And we defined two things. One is the shoulder. So this part of smaller RNAs that are closer to the marker. And then we found also a tail. And you can appreciate the difference if you compare it to what we regard as a good profile. And yes, and if you look, for instance, the profiles that are derived from samples, the FFPE samples, they show some similarity with this. And then we saw, well, this is showing signs of RNA degradation. And then we also found, and this I will discuss a little bit later, 27% also of this subset contain more than 5% of intergenic reads. And this could account for DNA contamination. And now I will go back a little bit in time with some of the results that Maija got before. So Maija had some data from RNA, from pairs of samples from urine. What she isolated RNA from urine, and then from urinary extracellular vesicles derived from that same urine. So she remembered that the profiles from urine were looking a little bit similar to what we were finding now with the profiles of women. And also something else, she had the sequencing results from these samples, and when we analyzed the data, we noticed that the samples that had this kind of bad profile had less map reads. So then they had less quality. They weren't so good for sequencing as the urinary extracellular vesicles or as the ones that have nice profile. And we found the same with our samples. So the samples that had a bad profile, and you can see them now on your right in the blue dots, had less map reads if you compare them with the ones that have good profiles. And the same happened for the samples that had more than 5% of intergenic reads. Those samples also had less reads mapped to the genome. So as a conclusion of this, we decided to exclude these samples. So based on the bioanalyzer profile, and also on they had more than 5% of intergenic reads. And well, now I want to give the floor to and thank you for your attention.
So let's continue discussing on the paper. So I would start with the main message of the paper. What is the main highlight from the paper? So we here show in the paper that there is a robust expression of more than 10,000 mRNA in the UEB samples in the population scale. And then if we do the kind of comparison with other tissues, so the kidney is the one which is most closely related with this expression profile. And in the disease case control analysis, we found differentially expressed genes which are enriched for the oxidative stress pathway. And when we derive a score based on a marker, a composite score, which we call transcriptional risk score, it was more correlated with the disease progression. So overall in this paper, actually we produced UEV profile close to 200 individual, which include technical variable, and main aim was to understand what are the technical variability in the sample and to study all these pre-analytic quality, which Karina already presented. And we know that now that we have a good quality data, then next aim was to understand what is the biological relevance of these transcriptome in terms of mapping with human tissue and comparing disease case and control. And this is the main schematic of the finding of the paper. So I will go through in detail in the presentation. So this is the content of the presentation as well as the paper. So I will start with a little bit background on diabetic kidney disease, then what was the study design of the paper, and then I will go to result. I will not discuss more of method because Karina already explained about the RNA sequencing, how we did it. And so we will go about what was the overall expression profile in the cohorts, because we did 3 cohorts, and then how this global profile looks in terms of human tissue map, and then what are our study subjects, and what is the clinical feature of them, and then what was the differentially expressed gene and how we did it, and then how those differentially expressed genes are kidney specific or cell type enrichment, or what pathway are enriched there. And ultimately then to see how those genes are correlated with more disease progression. So let's start with the diabetic kidney disease and its impact around 30% of diabetic individual, and even one of the major cause for end stage renal disease and major cause of death, diabetes related death. And its pathogenesis is partially known. One of the most accepted is that the hyperglycemia induced oxidative stress or stress in the kidney, which result also the development of fibrosis or accumulation, which progress for the more severe form of diabetic kidney disease. And etiologically, it's a polygenic disease. Many genetic factors are now associated with the diabetic kidney disease. The transcription factor also explored a lot, but the biopsies are rare, or there are no much biopsy. So this feature is less explored. So obviously there is one kind of overall aim is that develop more liquid biopsy. For this respect, UV is one of the interest for us. So what are the clinical marker for the diabetic kidney disease, which is used in the practice? So basically, to the appearance of the protein or albumin in the urine. And if you see with the disease progression, the occurrence of the albumin increases. And there are clinical cutoffs. So if there is a small amount, then is a start of the disease, microalbuminuria. And if you are in a more progressed form, then it's microalbuminuria. There are specific cutoff, and their second clinical marker is the EGFR, which is the creatinine clearance, and it's follow this curve where at the start of the disease you have a hyperfunction due to glycemia and kidney function more. So it's appear that the kidney is functioning more, but it's actually the start of the disease, and then the function goes down, and there are some individual who actually goes down more faster in this slope. And in terms of clinical unmet need in the biomarker space for the diabetic kidney disease, these are the one time point where people are trying to find new markers. One is the starting of the disease, where there is hyperfunction, no obvious albuminuria, but they may go to disease stage. So if there are more sensitive marker, and then also the marker which can tell that when the disease is, who are the individual who will go more down this slope more faster. Using non-invasive method and developing marker is one of the aim in this DKD field. And obviously we wanted to explore urinary extracellular vesicles mRNA. And so for that, we need many diabetic kidney cohorts. So there are 3-4 cohorts, and before that we also needed to understand what is the technical variability, and can we actually use this method in the clinical practice, like collecting the samples and transporting here, and if the expression profile is robust. So once we were sure about that in the technical variability analysis, which Karina presented, then And then we'll proceed for this two-stage design for to identify this diabetic kidney disease marker candidate. So we started with a discovery cohort of 72 sample where we actually compare the most severe cases, macroalbuminuric. If you remember from the previous slides, these are the most severe form of diabetic kidney disease compared to normal, and we identified differential expression. We did the qPCR validation, pathway enrichment analysis, and then correlation analysis with more granular clinical data, which are kind of causal factor for diabetic kidney disease like glycemia, HbA1c, blood pressure. And then we also try to understand how whatever the gene we found, how they are specific for the kidney tissue. And the pathway enrichment, we found is 6 mRNA, which involved in oxidative stress pathway. So we created this mRNA-based score to understand in a trajectory-based analysis how it's correlated with the disease progression. But of course, whatever we find here, we need to replicate. So we used at least 3 replication cohorts. So discovery cohort was basically mainly based on the men, because we know that male, female, there are differences, and there was also technical problems. So we started with the men, but whatever finding we found here, we try to replicate in the female sample as well. So let's go for the data result one by one. So this slide show what is the overall expression profile of UV mRNA in the discovery cohort. There were total 72 sample, and the first plot shows actually the number of the genes which with the count of 1 in each sample. So you can see that we detect more than 10,000 on average gene if we aggregate everything. And we took a stringent cutoff of more than 90% samples. The gene should be there for the common genes, because we saw that for the low expression, there is a lot of variability and we were not sure. So we stick with the stringent cutoff. And in this plot, you can see the distribution of the genes actually. So what is their expression level and what are their density, the numbers. So you can see this nice normal distribution of the expressions, which give us good confidence for the quality. And one of the main thing in the study design was that we have 24-hour collection cohort as well as only overnight collection cohort because of the limitation. So one of the main worry was that if we combine both, then could it be that there would be a batch effect. So this plot actually show the PCA plot for that. And you can see that there is no such clear clustering or there is separation based on 24-hour overnight urine collection. But there were a few outliers which we removed that may represent some kind of capturing other variations. So in general, we saw a good expression profile, and there is no other variable which explain these species. So we were confident there was no such global batch effect or that would explain this any kind of association. Then once we have this profile, then we try to understand in terms of doing the analysis to see from which tissue expression profile is UEB profile, which is in this matches most. So I took out the expression profile from the GTEx, which has a tissue expression data for more than 17 tissue, and where we have a good sample, like more than 50 samples to have good sample for this correlation analysis. And these are the tissues there. And if in the plot A, you see that our UV expression profile is the most closely matches with the kidney. And so it's kind of show that the variability, the co-expression pattern is much more similar to the kidney. So it's indirectly indicate that it might be the gene is contributed by the kidney. And then we thought if we remove out this kidney, then what are the other organ which is closest? So that was the prostate. Unfortunately, in the GTEx, we have very less urinary bladder tissue, only 9. So we couldn't do that, but we expect that will also be correlated here. And then further, we went out to use this deconvolution methodology and use only kidney-specific single-cell data as a reference and deconvolute, like what are the cell type we find in this UEB expression profile. So you can see that there is a good quantity of the proximal tubule cell, which I think kind of expected, because in the kidney transcriptome, proximal tubule contribute most. So we see same kind of mirror also here in the UEB. So this kind of give also the confidence that the expression profile has a biological relevance, which we expect. Then we went for the main differential expression analysis. So this is the study subject. So discovery, we compared normal versus macro, and you can clearly see they have a high albuminuria 24-hour or overnight, and low EGFR quite significant, and this are their distribution, but of course they are all men. And this slide show the differential expression analysis. X-axis is the p-value and y-axis is the fold change, and we took the most stringent bone for any correction threshold to restrict it to like most robust results. So we got like these 13 genes which are differentially expressed. And in this slide, actually, you can see what is the gene expression distribution of these 13 genes. So here, the macro were not involved in the discovery phase analysis, but you can see that in the most of the genes, the macro actually fit in the between the normal and macro, which is the severe form. So it's kind of show that the clear trend as DKD progress. So it is also indication that the expression profile are actually capturing the kidney dysfunction, and they are correlating with the severity of the disease. Then, of course, there was also question about the technical thing, because we did the RNA sequencing in different batches and 2 different places, and if the next generation sequencing is also kind of give some kind of technical variability which can affect this association. So we checked the association with the qPCR validation as there. So you can appreciate that in the actually the correlation of expression profile through next generation sequencing as well as from the qCR, and it's quite good correlation. Most of them have more than 0.8. So it's a replicate from the different methodology, and if we use the qPCR data, we again replicate the same kind of profile which we found. So it's good technical replication of the results. Then we went on to see what are the features which contribute to this association here, like diabetes duration, HbA1c, blood pressure are the main causal factor which is known in a field. And among them, actually, the most of the gene were actually correlated with the HbA1c. So here the significance is like for the most of them is significant up to 0.3. So that was what interesting observation for us, that it's a glycemia induced. It's also correlated with the glycemia and the DKD function in parallel. Then among these 13 differentially expressed genes, we focused more to see what is the co-expression pattern among them itself and what we can learn from them. So if we compare the same co-expression pattern using the real tissue expression data from the GTEx like kidney, prostate, and testes, so you can see that the expression pattern is more similar in the UVs if we compare to the kidney compared to the prostate and testes. Like there are some genes like here MAP7 and TMA9 which are not very specific to kidney. They are also in the prostate as well as testes. So in that sense, it was good to show that this correlation is much more similar to real kidney tissue compared to the other tissue, which kind of also indicate they are coming from the kidney. And then finally, we went to databases where there is a single cell sequencing data and what is the proportion of the, what are the fraction of the cell which these cell types express for these genes. And you can see that so as we know in nephron, there are proximal tubule, distal tubules, and in the glomeruli, there is a podocytes, and then endothelial and mesenchymal cells. So among these cells, you can see that the most of these genes which we are able to find in the database, they have highest expression in the proximal tubule cells. So based on these observation, then we manually checked what are the genes, what are kind of the function these genes are involved, what the function of these proteins. So out of those 13, the 6 were involved in the kind of oxidative stress, either like in a H2O2 pathway or ammonia-related stress pathway, or chaperone, which is involved in the protein protection during the oxidative stress. So given that oxidative stress and glycemia is one of the main factor which is shown by other studies. So we use these 6 genes and then we constructed a transcriptomic score based on the average of the expression of them, normalized expression giving equal weight. And so here this figure shows this stress scores. This is discovery cohort where macro has a most stress score compared to normal, and which is expected because the differential gene expression come from this cohort, so it's expected. Then we replicated this in 2 cohort in a female T1D cohort, then type 2 diabetes cohort, and then there is third also replication cohort. So in the combined replication cohort, which is the E, you can see that they are same kind of trend as a discovery. So it's nicely replicated our observation about the increased mRNA level of these candidate gene. Then we also use some negative controls, like if this stress score also correlated with only diabetic versus non-diabetic. So there was no such correlation here. It's not differ that much between male and female. There was same kind of distribution, and the people who are using the medication, so without medication, no medication, there was no difference. So these negative control also kind of say that whatever the stress score we are showing, it's related to kidney dysfunction. It's not very unspecific, like only with the glycemia or medication use. So finally, at the last, since this was quite a detailed cohort where we have eGFR data for to the 14 years, so we can derive this longitudinal trajectory. And it was also important to see how this expression profile, which is only one time collected, how it's correlated with that trajectory. So overall aim was that if we can predict this longitudinal trajectory based on one time point measurement. So this is the discovery cohort. Here I divided the individual based on stress score quantile. So this is the highest quantile, and then these are the other quantile. So you can, of course, expect that the individual with the highest quantile, which is mostly macro, they will have this kind of like faster decline. But then we actually subdivided our normoalbumic, which are non-decayed individual, and what is their profile. In the lowest quartile of the stress score, which is up to 25 percentile and above that, and compared to one positive control, which is the macro, which is the early decay indication, and how their eGFR decline look like. So you can see that the normal individual, which are healthy, their decline is much more similar to macro if we divide based on this stress score. So stress score able to tell that among the normal individual who has a higher expression of these genes, their clinical history of the eGFR decline is much more similar to the macro, which are early clinically confirmed DKD subjects. Then we replicated the same pattern in the T1D female samples, where the individual who are in the lowest quartile, they have almost stable eGFR throughout these years, but the individual who have a more than 25 percentile, they have a decline similar to the micro, which is early. And then the replication 2, so replication 2 is also important in that sense. These are the DKD individual, which are macro and micro. And in the macro, most where they are in the highest quartile, but there are few which are like less than 75, and we divided into 2. And you can see that actually this, in the macro, which are in a highest quartile, they have a steepest decline, but even the macro, which supposed to be like higher decline, the individual who has a less stress score, their decline was really under the -1 per year compared to the -3 here. So there is a significant difference based on the this stress score stratification, and the same we see with the micro as well. So then we also replicated this if using the quantitative trait and correlation. So it was quite nicely correlated. So this is a score not only associated, but also associated with the clinical trajectory of the decline of the eGFR. So could be a good further marker to development as a like faster decliner or early early DKD progressors. So these were the main results. So conclusion is the same as explained in the our starting slide. So more than 10,000 mRNA and kidney similarity with the kidney tissue, as well as the genes which we found make biological relevance for the DKD pathogenesis, and it's also correlate with the disease progression. So of course, one of the main aim of the study also was to build the resources. So we actually built this website where you can actually check the gene expression of your favorite gene. Like here I put, and it's kind of like this site will be developed as a kind of public resource. Although this data is already present in the paper, but it could be good to explore this site. So there would be like much more data harmonization in that respect. And of course, the future plan is to replicate the finding and more special. And now, actually, Maija will explain a little bit more on the future plans.
So thanks, yes, indeed, we want to keep exploring the messenger RNA markers of the DKD in more focused groups, and also doing a meta-analysis, because we still have like hundreds of samples left for analysis. And we would also like to understand the spatial expression within the kidney tissue, whether it gives us further information of the pathogenesis mechanisms, and looking at those stress score markers or other markers. Then we also want to keep developing the eVRNA quality control, and here we have a collaboration with Dr. Lassi Paavolainen here at FIM. And we also want to keep developing scalable methods for EV and secretome isolations and biomarker analysis, and we do it in our hybrid core, but also in collaboration with Dr. Pia Siljander. We have this FAST EV project and other collaborations, for example, with Dr. Janne Leivo, Carmen Geronimo, Genevieve Part, and Seppo Vainio. And last but not least, I would like to mention that I've been really happy to notice the activities in the urine task force and also in the GUSEV, and I think these are fruitful platforms for further collaborations, develop these scalable methods and biomarker analysis. So with that, I would like to thank for your attention, and we are happy to take any questions.
Thank you, Maija, Karina, and Prakash, for sharing your work with us and for the engaging presentation. Congratulations on your amazing and impactful work. So we're going to allow unmuting now. So if you could put your questions in the chat box, and then we'll call you in order that the questions come through. So we'll start with Edgar with this question.
Yes, thank you so much. So I had a question about the read. So at the beginning of the talk, you mentioned that you were excluding samples with many intergenic reads, and I was wondering why would you do that, since that is a big part of the whole content of UEVs?
Well, it's expected that the intergenic reads come from the DNA, and we wanted to do like RNA analysis. So this is why we kept it as a quality criteria to exclude those that had too much DNA contamination.
So you really only wanted to focus on the mRNAs then?
Yes.
I see, that makes sense.
Elena, your question is next.
Yes, again, congratulations, everyone. Really very nice work. I was puzzled a little bit by the data that you showed for the eVRNA quality controls, and particularly those 27% female and 1% male that were excluded. And I may have missed that, but did you actually see if that correlates with the EV number? Because what we've seen in Rotterdam is that often females tend to have a bit more lower EV count and more diluted samples in general.
Maybe I can answer this as well, and others can comment also. Well, we didn't measure the particle numbers from all of the samples. However, what we do is that we apply the same RNA quantity to the library preparation and sequencing. And there we didn't see that much difference between males and females. And I wouldn't think it like the lower number of EVs impacts the quality, like having more fragmented nucleic acid species in the sample. So
no, that's not what I mean. What I mean is like if you have a low RNA concentration, and you could see on your graphs also that the good samples, they were at about 100, while the other ones were at about 20. So you have like 5 times difference in RNA quantity there to start with, and then you actually really zoom on those smaller fragments and the fragmentation. So what I think is you're looking at is not so much quality differences, but rather quantitative differences. And because the bioanalyzer tends to auto scale there, you are actually not having that much poor quality, but actually low quantity samples in those 27 plus 1%.
it's not so in all cases. Sometimes we have huge quantity. Actually, that's one of the markers we use. If we have too much RNA coming from the samples, is sort of a sign for us that there was probably a cellular contamination also. So in those cases, we can also see this fragmented pattern. But you're right in the sense that if we have very low yield of the RNA, it's difficult to actually see how does the RNA pattern look in the bioanalyzer profile. So in that sense, I think you are right that those low yield samples are challenging.
Yes, it is. Okay, thank you, Maija.
Phil A had a question.
Yeah, this is a tremendous amount of work, and it's quite surprising to find so much mRNA in these samples, but it's disappointing clinically, and I am a clinician. A kidney biopsy is the gold standard, and this doesn't seem to be much better than that, unfortunately. And I was wondering if a microRNA or other RNA might yield more definitive. Stress is nice, but it's not going to help me as a clinician.
Yeah, that's of course, good point, like how specific is stress for the diabetic kidney disease. Could be also that we get the stress signal from other kidney disease, other diseases. But I just think we need to keep exploring the other messenger RNAs. Maybe we find more like cell type and tissue type specific signals. So
that would be very nice to know what cells in the kidney the mRNAs are coming from.
Yes, of course we have
Which
indications.
is a very complicated thing.
Yeah,
but
our
wonderful aim.
proximal tubular cells. So that's at least suggested by the single cell sequencing analysis and like kidney cell type deconvolution analysis. But I think we get more information when we analyze bigger cohorts of patients. So maybe we also get to correlate to different kinds of clinical parameters that we did not explore yet.
Yeah, maybe one could be think that when we have these biopsy data where we have a real kidney expression data, we can correlate with that, how these UEB expression data correlate with the real expression data, and if we
I admire your persistence.
Yeah, if we have more granular phenotype like histological information and then try to correlate of these data. So it could be one way. So basically we need to explore more, replicate and more granular phenotype to see if these signal further enriched. Yeah.
I have a quick comment for the community. So I am a 1-year-old member of ISEP, and I work on all of these bioinformatic analysis for EV data. And my question to the group that presented is that you may not look into some of the data sets you have for urine in the xRNA atlas, and also whether there is a task force specific for RNA or omic analysis on EVs.
Sorry, is this question to us or generally to the urine task force?
The first one is to you about whether you managed to take a look at the xRNA Atlas database.
No, not yet, but it's a good idea. Thank you.
And the second question was to the community whether there is a task force that specifically approaches how to analyze omics data coming from EVs.
We do have a working group within the urine task force that is particularly focused on normalization of omics data of urinary EVs. So that is the closest one that it comes to what you're asking. But there's always possibility to open a new task force or to start a working group within the existing ones that can focus on what you are interested. Point that back to Maija because she's now in the leadership of the task force as well. So we can pass the message and discuss that.
Thank you.
The next question is from Sam Dash.
Oh, sorry. I thought we were just going to read the question. Anyway, great presentation. So my question is for Karina that you showed ring numbers, vary from 2 to 10, and I was little skeptical whether there is any difference in time you kept the urine in room temperature versus you put it in -20 immediately or -4 degrees or something, or maybe the way they collect is different than the other sample which has a better ring number.
Sorry, there was an echo because we had two computers in the room. So yes, I mentioned the ring doesn't apply to the RNA derived from extracellular vesicles. And I just mentioned it because to give a background and to say that we don't have such an index now to evaluate the quality of the RNA in our extracellular vesicle samples. So we just send to sequence the samples that we get, but it could be good, and as I mentioned, to have an index to filter the samples before sending them to sequence. So that's why I mentioned the ring. Regarding that, why the strange profiles that we got, we think that is the type of sample, specifically women samples have this issue, not the main derived samples. So yes, it can be because of the collection and how the sample is collected. And of course, we think that the handling before freezing, like for instance, the pre-centrifugation step before freezing can also be an important factor impacting the quality of those samples. But Maija, maybe you have something extra to comment regarding this?
Okay, so I was just wondering whether it was clear that we don't get ring 10 values from the urine EV samples. So that was just an example how the typical good quality cellular RNA is looking. What we typically get from urine EV samples is around ring 2, but ring is not that relevant for the urine EVs because of lack of ribosomal RNAs. At least they are not in that quantity as in the cellular samples.
Yeah, I can comment that we also see exactly the same profile from urine in New York Mount Sinai samples. Mm-hmm.
Okay, the good quality profile, you mean?
Yes, although the core hates us because they say urine is not adequate, so we have to spend lots of time explaining that that's how it should look like.
Okay. Yeah, it's quite typical, but if we sequence more and more urine or EV samples, then they get used to them.
May I ask which technology you're trying to use for spatial analysis and whether it's going to be at the EV level or cell level?
Okay, Karina can take this one.
Yes. Sorry, can you repeat the question? Is yes.
Yeah, what spatial technology are you planning to look into or develop for your work?
To isolate the high throughput vesicles or to assess the RNA quality?
on your future work, you had a point which was what is the future, that you were going to look into the spatial expression of the mRNAs. So is this going to be for tissue?
Yes.
Yeah.
Yes, exactly. Sorry. So we want to well, we have maybe 2 ways to go. One, of course, is targeted spatial genomic, where we only check the expression by in situ of these targets that we found. And another approach is, for instance, using technologies where you can screen thousands of messenger RNAs, also by in situ. But this is, well, I don't want to give the name of the brand, but yes. So it can be. I will go first with the targeted approach because we really need to know which cell types are from where is coming the signal that we got, the stress score. But of course, it would be nice that once that we have the biopsy to do some global analysis of the different messenger RNA.
That sounds great. Thank you so much.
So the next question is from Uta, who's had to go. So I'll read her question out. So really great presentation and work. I'm surprised that you do not find more signals from the glomerular part of the kidney. Any comment?
Maybe I can start and Om can continue, but we do find some signals from the glomerular part as well. However, we were focusing on the most differentially expressed genes, and those happen to be derived from the proximal tubular cells. But maybe in the future analysis, we can concentrate more on the podocytes and that department.
So in the paper, we focus most on the most stringent candidate gene. But in general, if we try to look the glomerular specific genes, so they were quite expressed in the UAV. So they were, I think, remember around 200 such genes. Out of that, I think 30-40% were there in the UAV. So they are quite highly expressed there, but they are not that differentially expressed in this set of samples which we use for comparison. But maybe if we increase the sample size and do more kind of analysis, focused analysis, then maybe we might find the glomerular specific gene as well. But there were indication like below the genome wide threshold, there were some genes. But of course, I didn't mention due to this monoclonic correction, but there were.
And next question is from Genevieve.
Sorry, hi Maija. I was wondering, what evidence do you have that the intergenic reads are really DNA? Because they could be long non-coding RNA.
I think one point is also RNA sequencing is, I think, oligo-T. So we expect to have like mRNA only, not like whole RNA content sequencing. So, and if we are finding that intergenic reads, so which we do not expect based on cDNA, then it may be indication of contamination or something kind of quality problem. So I think
do you actually do DNA digestion to verify that they disappear?
In this case, we did not do the DNA treatment, but generally those like intergenic reads, the higher proportion of them went hand in hand with the occurrence of those like bad profiles, giving the idea that there are more of these fragmented nucleic acids in those samples. So I think it just generally proves that the samples were not all right. And it would suggest that also the intergenic reads came from the contaminating DNA. But you're right. Nowadays, we have started to use the DNA treatment. Initially, when we started with the male samples, we had no problems whatsoever with this DNA or intergenic reads. So the percentage was really low. It was like, I think under 0.1%. but later on exploring more cohorts and these female samples, we found that those seem to get more of the intergenic reads.
So we're over time, so we're just going to finish off with one more question from Benedetta.
Yeah, okay, thanks. Very nice work. And I think it also underlines the fact that proximal tubular cells are a very important target of diabetic nephropathy, which is very important. But my question is more related to your cohort with macroalbuminuria and increased permeability. So this is a good chance to evaluate the possible presence of also serum or glomerular derived EVs in the urine. So could you speculate on that? Did you see any signature from serum or endothelial cells?
So for the principal component analysis, we used whole of our type 1D discovery cohorts, also including the macroalbuminuric individuals. And from those plots, as you could see, they clustered quite well together and not close to the blood, which was one of the tissues from the GTEx. So in that, it would look that even though we had patients extending from normal albuminuria to macroalbuminuria, there was not such a great difference in their RNA profiles.
And I think in the paper, there is also one analysis where we wanted to check some simple straight check, like if the genes which are reported to be not expressing in the kidney, there are 6,000 such genes in the human protein atlas. And what is the number of that genes which are detecting the UAV? So we detected only 200, only 4% out of those 6,000. So in that sense, like the EV from the other circulation might be not that much prominent in the urine at least. Like,
and did they increase in macroalbuminuria?
No, no, it's remained constant.
Okay.
6 to 7%. Yeah.
So thank you, Maija, Karina, Prakash, for sharing your work and your fantastic presentations. We wish you all the best on your further work, and we look forward to seeing more work on how sort of EV cargo can be leveraged in kidney disease. Thank you to everyone who joined today and all your good questions and discussion. Hope you have a great festive season and a happy new year. And we look forward to seeing you at EV Club in the new year.